Hello, everyone, and thank you for joining us for our final presentation of the day. Our next presentation is on genetics and CMT, presented by Dr. Stephen Zuchner. Dr. Zuchner serves as the chairman of the Dr. John T. McDonald Foundation Department of Human Genetics and the co-director of the Hussman Institute for Human Genomics. Dr. Zuchner obtained his medical degree from RWTH Otkin University Medical School, where he also completed residencies in neurology and neuropathology, and his, quote, Dr. Med degree. Dr. Zuchner completed his postdoctoral studies at Duke University Medical Center in Durham, North Carolina, where he subsequently achieved assistant professor status. In 2007, he moved to the University of Miami as one of the founding faculty of the John P. Hussman Institute for Human Genomics. In 2015, he received an honorary doctorate degree from the Semmelweis Medical School at University of Budapest, Hungary, for his contributions to next-generation sequencing and international studies of rare diseases. His research interests are focused on identifying genetic variation associated with disease. His lab is internationally renowned for being one of the most successful in identifying several dozen genes. His group is among the pioneering groups that have promoted genome sequencing methods for disease gene identification in humans, mice, worm, and Drosophila by utilizing in-house developed software solutions. He is currently pursuing large-scale exome and genome analysis in multiple neurodegenerative disorders to map their complex genetic architecture. These activities had led to multiple patents and translational assets. Therapy applications based on gene editing technology are currently also being developed by his group. So Dr. Zuchner, I'll turn the symposium over to you.
Can you all hear me?
Yes, we can.
No, you can hear me. Good.
Yep.
Well, thank you for the introduction, and I'm very glad to be here. I'll talk about the genetic aspects of Charcot-Marie-Tooth disease. As you know, CMT, which is a peripheral neuropathy, is a genetic disease, first of all. And you probably heard already from Dr. Shai how we divide this disorder into different types of sort of genetic diseases. Like here under this umbrella, we have the dominant, the X-linked, and recessive forms. So I also want to say before I dive into everything, I'm gonna explain to you a little bit what genetics means in CMT and peripheral neuropathies, and tell a little bit sort of science 101 genetics, and then sort of dive a little deeper. And I hope you can follow and sort of enjoy where research is going nowadays. And so yeah, maybe next slide. Next slide. Okay, so this is really just to illustrate how the genetics of CMT, which started out with basically one discovery in 1992, has now about 100 genes that can cause the disease. 100 different genes, and each one can cause quite similar, what we call the phenotype. So for patients, it's very important to understand which particular one of these 100 they belong to. Next slide. So really starting with the basics, when you think about your body and our bodies, it's actually a sort of aggregate of some 40 trillion cells. So these are individual cells, and about 100 billion make up the nervous system. And the nervous system also includes the spinal cord, of course, brain, spinal cord, and also the peripheral nerves. And the peripheral nerves are the ones, the cells that are damaged in CMT. And that's why patients have weakness, foot deformity, cannot feel so well, and so on. So what is astounding is, from a higher level, is that each of these billions and trillions of cells, each one has actually a complete copy of the genome, of your personal genome inside. And human genome is about 3 billion letters long, and you have 2 copies. So 2 copies of these long letters in each of these billions and trillions of cells is quite astounding. And what is also quite a sort of interesting side aspect is that all life on Earth, plants, bacteria, humans, we all share the same genetic alphabet. So the genome is written with 4 letters, A, T, G, and C, instead of the letters we have in our alphabet. So it's less letters, but billions of them. So here is an illustration of a cell. Let me see if I can have a pointer here. Can't do this, I guess. First, yeah. So you see a cell there. It's also written in there. Can you see my cursor? Does this work, Nicole? Yeah. And then within the cell there is a structure called the nucleus, and in this nucleus there is the copy of your personal genome, and this is sort of a magnification as it comes out of it. We call this a chromosome. So the genome is so big that it has to be packaged, and the way it's packaged is called chromosomes. These are individual bits of the genome, and when you untangle these chromosomes, you eventually end up with this stringy structures here on the right side, with these what we call nucleotides, or sometimes base pairs, the G, C, A, and T here in color on the right side. So this is how it looks like, sort of ultra microscopically, and this is what we study in genetics. When these nucleotides are wrong, then you may get actually CMT. Next slide. So what is known for a long time, of course, is that in CMT and other genetic diseases, that you often find a family history, that not just one family member has the disease, but multiple ones. And by studying how family members also have the same disease, we came up with different versions, basic sort of versions of how inheritance works. And there are 3 main ones: the autosomal dominant, autosomal recessive, and the X-linked forms. And I'll go into this in the next slide, please. Next one. So a very important form is the so-called dominant inheritance. In a sense, so in the schema, to explain it, is you see their father and mother. As I said, each person has 2 copies of the genome in each cell. So on the father, you have 2 columns. They represent the 2 copies, same for mother and also the 4 children. In a dominant disease, one of the 2 copies has a, what we call mutation, has an error, so to say, in the genome. So one of the 2 has an error, and in this case, the father has it, and then the father gives it down to the children, right? So what really genetics is all about is how we inherit our genomes from our parents. So we get half of our genomes, like 1 blue column, from our father, and the second one we get from our mother. So we all end up with 2 genomes again. And then we pass down those genomes, 1 copy of it, to our children. So in this particular case here, you see how the father hands down 2 of these wrong copies to 2 of his children. And that's quite typical for dominant, that about 50% of children do get the disease. So of note, again, a parent has the disease and also a child has the disease. That is typical for dominant. Go to the next slide. So here, this is what a pedigree would look like. Again, there's always a sick parent and a sick child, and then the sick child can become a parent and passes it on. So this kind of pattern, again, is called dominant. Now, different is when we have a recessive disorder. In a recessive, actually, the father carries a wrong copy, 1 copy, and the mother carries a wrong copy. However, both parents are healthy. They don't have the disease, even though they carry a wrong copy. So we call them carriers. Now, if a child by chance receives the wrong copy from the father and the wrong copy from the mother, then this child will actually develop CMT. So to develop CMT under a recessive model, you need 2 wrong copies. And when you sort of work out the chances here, then about 25% of children, so 1 in 4, would receive 2 copies. In real life, however, that doesn't always play out so well. It's more like rolling the dice. Sometimes you get a 6 twice. And then in this case, they would have maybe 2 affected children. Importantly here, when you go to the next slide, or maybe another one. In a recessive disease, both parents are healthy. It's very unusual if they also have the disease. So, and children have the disease, if you have a family history that looks more like this, where both parents are healthy, and you may have CMT, and maybe one of your siblings has CMT, that's most likely a recessive form. And there are different types of genes that we look for the different forms. So really what happens in practical terms when you go to a doctor, of course, the diagnosis is made, say at a neurologist, and then nowadays you would order a gene test. In the past, we have done so-called single gene tests with a technology called Sanger sequencing. And what we do today and for the last decade really is we do multi-gene testing. And the technology is called next generation sequencing. So in a sense, you don't test just one gene, but in the best case, you would test all 100 CMT genes in one go, and then hope that one of the ones will be, be your diagnosis, and then you would get a definite diagnosis, and the exact gene you carry. Other forms of sequencing are so-called exome sequencing or genome sequencing. The exome sequencing is where we sequence not just 100 genes, but all 20,000 genes. So we have about 20,000 genes in each person. And then genome sequencing is sort of the absolute maximum you can do. That is when you not just look at genes, but you also look at the surroundings of genes, at the space that is in between genes. You try to sequence really the entire genome. Next slide. So here's a bit of an illustration of that. It all comes down in the end of the diagnosis, a single mutation, a single change, sort of on the left side of at the tip of this triangle. To find this, nowadays you would do so-called gene panels. So you test multiple genes at once. And often today, because the price is really dropping, you don't start with small panels. You often go into so-called bigger gene panels that test all the genes that are known for a disease. And if you still cannot find a disease gene, then you would potentially go to exome sequencing, which is a much more complicated test, or the entire whole genome sequencing test. Next. So yeah, again, exome sequencing, much more involved. This test actually is only around for about 10 years now. And we have seen a lot of discoveries because of this test is now available. Sort of what are some of the complications for this test and also gene panel tests is that because you sequence such a big space of the genome, basically millions and billions of bases, by chance you actually find variants, you find changes inside of CMT genes and outside of CMT genes that may cause a disease, but you can't quite be sure because maybe these variants have never been seen before in any other test. Next slide. So these variants where we don't quite know what they mean are called variants of uncertain significance. The word says it's actually what this is. So these are changes in the genome in a person that are neither benign, because there are many benign changes, but they also we are not certain if they're pathogenic, if they cause disease. So we call them sort of uncertain significance. These variants have become quite a problem for diagnosis. In a recent study of some 25,000 patients that underwent testing, about 50% of these people had such variants of uncertain significance. And what happens really is when your doctor receives a report back, he's really not in a good position to tell you if this variation explains your symptoms or not. So in a way, we do the test, we expend the effort, the money, but we still don't get a good answer. And it's very dissatisfying, really. So there are a lot of efforts that go into trying to tease out which of these variants of uncertain significance are actually benign and which are truly disease-causing. Next. And here again, just some nomenclature. A positive test is often called a mutation. So if somebody has a mutation in a gene, that typically means it is causing a disease. In this case, let's say CMT. If the report is negative, then no mutation was found. There may have been some benign variants that don't cause disease. They just there by chance. And all of us, we carry these variants that do not do much. And we can say with certainty, they are just benign, they're negative. And then there's this fairly large group of uncertain significance. Thanks. Next one, please. So you can imagine that the doctors who specialize in this, they have come up with quite elaborate schema to decide which of these many variants we find in exomes and genomes, which of those we should call benign, which we call pathogenic, which ones are mutations. And here's just one of the most important of these schemas called the ACMG guidelines for variant interpretation. And I just show this to you as that you see sort of the tools that the doctors and geneticists work with. Next one. So beyond sort of understanding which variants matter for CMT, there are other challenges for the classification of CMT. And here's one example. There's a gene called GDAP1 that is known for a long time to cause CMT. And it's just one example that this gene actually has been shown to cause CMT as a recessive disease. And in some cases, though, it can also cause CMT as a dominant, in a dominant fashion. So that makes it even more complicated to understand variants in this gene. Typically, in recessive, again, you would expect two variants, two defective, so both copies of the genome have a mutation. And if you have only one copy, you would say, well, this is not a recessive, so this cannot explain this patient and this gene. But this is a gene where you can have actually both. And it's very hard to decide sometimes which of the variants are dominant and which are recessive. Next. Another complication is that we have found over the years now that what we thought are CMT genes are actually also, these genes sometimes cause other disorders. Or patients who carry these genes, they have maybe both disorders. So each circle is sort of a disease, a neurological disease, and you see the overlap of these circles sort of illustrates how how these diseases are not completely separate, how the genes are not separate, but how they overlap with a broader phenotype. And again, that also can cause challenges. If genes carry variants, and these genes are, say, known for spastic paraplegia there in green, you still wonder if maybe they also cause CMT. Next. So to sort of sort this situation out more systematically, and remember there are 100 genes right now, so it's quite a task. We have developed this variant browser, this neuropathy variant browser, which is an online tool. And this is a place where we sort of collect from the literature, from clinical laboratory tests, we collect information on these variants that have been observed. So there are thousands of these variants here in this variant browser. And doctors, sometimes also patients, can go here and check if a particular variant has ever been seen in another CMT patient. And the nice part about this is being used not just in the United States, but also in Europe, in Asia, in Australia, and so on. So actually by comparing CMT mutations from different continents, we found this is a very powerful approach to work out which variants cause CMT and which ones do not cause CMT, even though they all fall in the same gene. So so you think of like 100 genes, well, that's probably it. Unfortunately, what turns out is that even with all these genes, and when we apply these big tests, only about 50% of patients get a diagnosis today. So this is the unexplained heritability. Sometimes we call it the diagnostic gap. And it is a big problem because a lot of patients who clearly have CMT don't get a genetic diagnosis, which means in the long run, these patients will not really have access to these new kinds of therapies that are coming now, which are very gene specific. So that's a big effort we are all involved in, is to identify the genes and mutations that sort of fill the remaining gap. Next. And luckily, technology, especially also developed here in the United States, has really improved so much in the last decade that the cost for these kind of studies and also the cost for these tests has dramatically fallen. So we can actually now do genome studies on thousands and thousands of patients for a cost point that's doable. That would have been absolutely not possible just 15 years ago or so, when a genome would still cost millions of dollars. Next. So, and where we also involved in, and this is not just us, this is supported by the different patient organizations. It is supported by the Inherited Neuropathy Consortium. We have developed these online tools that bring together these data, the exome and genome data from CMT patients. This involves many de-identified patient records and many researchers from different countries. And we sort of look together at this data continuously and with the goal to find additional genes to close the diagnostic gap. And this is just a screenshot of one of these tools called the Genesis platform. Next. So here's just give you some taste. These are countries that involved right now that have sort of sent data to this database. And over the last 8 years or so, some 70 novel genes and about 25 of them are CMT genes have been identified, have been discovered using this approach, this platform. And it's very popular in the medical literature, and there are now 11,000 data sets. It's really becomes a big data, very computationally challenging undertaking. Next. Just to give you an idea here, again, this is a discovery we published about a year ago. It's a novel CMT gene called ATP1A1. And the way it was really discovered is with this Genesis platform, we had united data from Dr. Shai in Iowa, but also collaborators in Czech Republic, in southern Italy, but also in South Korea and from Australia. And only because we had all these data from the different scientists, we could actually see a pattern in the data, a signature that allowed us to find a gene. If we had only worked with Mike Shai, for instance, we probably would have not found this gene. You need input from different regions of the planet and multiple investigators, really. Next. So here's a list of genes. These are just the genes discovered in the last 8 years. And you can see how there's simply significant progress and in finding out what is the genetic architecture or the genetic basis of CMT. Next. And another aspect that researchers are very interested in are so-called modifier studies. They are also very challenging studies. The idea here is really that we know for a long time that patients, CMT patients who have even the same mutation in the same gene, they may have very different onset of symptoms. The severity may differ and other elements of the disease. So there has been this idea that surely the environment for each patient matters, like your diet, the place where you live, and other factors that certainly modifies how your disease develops. But the idea is that there may also be secondary so-called modifying genes in people that sort of determine if you have an early or late onset of symptoms. And the hope is that if these genes exist and we can find them, maybe it's easier to sort of have a therapy for these modifying genes. If we could, for instance, make a disease onset, move it to later in life or make it milder overall, that would already be a big success. So we have set out to find these modifier genes. And these are some details I probably don't want to go into too great detail. Next, please. We did what is called a genome-wide association study in CMT patients, and we found that one gene there on the left side called Cepa1L2 has reached our threshold for testing. And we think it's the first modifier gene for CMT. Next. And what we also found is that this gene, Cepa1L2, when we test this in cell cultures of Schwann cells, that it actually suppresses how other genes that are known to be important for Schwann cells, how they are also suppressed when Cepa1L2 is suppressed. And next. So why is that important? Potentially, that could mean that if we had a medication, a drug that can suppress Cepa1L2, which is a modifier gene, it does not cause by itself CMT. We can actually reduce, for instance, the amount of PMP22. It's the gene right next to it. And PMP22 causes CMT when you have too much of it. So this could be an example where if we have a therapy for a modifier gene, we can actually influence a gene like PMP22 and maybe have a therapy that way. Next. And one more discovery here, and I know that it's going to get very scientific here. I hope you're still with me. This is a very recent, very exciting discovery. In fact, what you see on the left side is the cover of a scientific journal called the Nature Genetics Journal. It's a very, very high-end journal. And you can see that this particular paper made it on the cover of Nature Genetics. So it's definitely considered also by the scientific community as a breakthrough discovery. So what is it? In a short sense, we found that the so-called SORD gene, S-O-R-D, is a novel CMT gene. And the special thing about it is A, it looks to be the most common recessive type of CMT, and B, it's potentially the first treatable CMT form that we know of. We're very hopeful about this. So I want to lead you in 3 slides through the discovery, which we hope will have some impact on patients' lives very soon. One back, please. So first you see sort of in the middle of this slide that there's sort of the structure of this gene or protein, and you see the mutations we found in this gene in different patients. And then below you see sort of typical pedigrees. And you probably remember when you see like the family tree, family pedigree on the most left one, that this is a typical recessive pedigree. Both parents are healthy, but they have 2 children with CMT. So parents healthy, they are carrier, they carry each a one mutation, and they give it to the children who then end up with 2 mutations. And only when you have 2, you get the disorder. So recessive CMT, when you look at the pedigrees in the middle, what you notice is that everything that's sort of a black box or black circle is a CMT patient, that these 2 pedigrees show only 1 patient in the family. Now think about it. If you would be that patient and your parents are healthy, your siblings are healthy, your aunts and cousins are all healthy, that is what we call a sporadic patient. And that makes it very difficult sometimes for neurologists to work out what type of neuropathy do you have. Do you have really CMT, or do you have a inflammation in your nerves or something else that harms your nerve, like diabetes can do that. So being a sporadic patient is especially difficult for diagnostics. And we're very happy that we have that this new gene can explain many sporadic patients that may not know or have given up the hope that a source of their disease can be identified. So when you go to the next slide. Here are more examples. These are all the families we have in this very first paper. And again, you see the typical recessive pattern in many of them, but actually at least half of these pedigrees, if not more, you have these sporadic situations where there's only 1 person in the family that has the problem. So it's not so clear for these families. Is this genetic or is that not genetic? Next. So how does this work? Again, in this sort, we have the very unusual situation that we were able to not just find a gene, but at the same time work out what is wrong exactly on a molecular level and what can be done about it. So first on the left side here is the so-called polyol pathway. Strange word, but what it is this is sort of a side pathway of how we deal with glucose in our bodies. So glucose is the most common sugar. Nerve cells depend on it. Sometimes when we have too much sugar, which is called diabetes, the body tries to get rid of too much sugar. And one way is to activate the polyol pathway, sort of a pressure valve for for too much glucose. And what happens then is the glucose is being changed by the sort of yellowish box there. It says aldose reductase. This is an enzyme. So this enzyme takes glucose, then gets activated, takes glucose and turns it into another molecule. By changing it slightly, becomes another molecule from a sugar into what we call a sugar alcohol. And that is called sorbitol. Now you have may heard of sorbitol. It is also a quite common sweetener artificial sweetener for coffee or tea. So sorbitol is also known when you have too much of it. It is not good for you. It actually damages your nerves. This is being studied for a long time. That is why your body would immediately activate this purple boxed enzyme called sorbitol dehydrogenase and would remove the sorbitol. It would change it from sorbitol to fructose. So fructose is a fruit sugar. It's water soluble. There is no problem to deal with this in your body. But too much sorbitol is not good. So sorbitol dehydrogenase, in fact, is the long word for SORT. So this is the very gene, the enzyme that is defect in these patients. So when you don't have sorbitol dehydrogenase, what happens? You end up with too much sorbitol in your body and in your nerve cells. And it seems that nerve cells are especially susceptible to too much sorbitol. Next. And in fact, when we looked into these patients' blood, we saw that normally controls there, you can almost not measure sorbitol in blood. It's almost not there. In patients that do not have the SORT gene, it's over 100 times higher the level. And so that is definitely not good for you. Next. So we also asked patients to give us a skin biopsy, a small sample of their skin, and so we can grow those skin cells called fibroblasts, can grow them in a petri dish. And I hope you can follow me here. The left graph or plot, you see the first three colorful dots there. These are from normal patients. We call them controls. And you can just see they all hover around 0, okay? And then the next three are from patients, from SORT patients. And the first one, the purple one, is higher. It's about 4 there. This is the level of sorbitol that these patients have in their fibroblasts, okay? Way too high, not good. Now, excitingly, when we give 2 drugs to these fibroblasts, the level, the blue and the green on the right side, they come down to almost 0. They come down to the normal range. This is because we gave these drugs, and they called apalrestat and ranirostat. And so we have actually shown here in patient cells that we have effective drugs in our heads to normalize the sorbitol level. So then we did the same in a fruit fly genetic model. Now, this may sound crazy to use fruit flies, but it is definitely a way to save mouse models. And it is also much faster. You can make faster progress than mouse models. Eventually, we'll have to do a mouse model, but starting with fruit fly is a good idea. Turns out you can do a lot of interesting studies in fruit flies. And these fruit flies, by the way, when they don't have SORT, they get a form of CMT. And just to shorten this here, you can see the same effect when we feed these fruit flies. When we feed them these 2 drugs, they basically normalize their CMT, so they don't get CMT. Next. So that's basically the SORT story. And just to say one more word on this, I'll be very hopeful that we're already talking to different companies that we can develop this into clinical trials over the next year to make these drugs available to CMT patients in the United States. So I want to switch gears a little bit towards the end here and talk about family planning options. Today, it's true that we don't have a therapy for any type of CMT, okay? And so there are parents or families that do consider family planning as an option. They just don't want their children to have CMT. It's not something that works for everyone, but for some families, they definitely look into these options. Basically, preventing their children with technical methods to not have CMT. And the most common really is that you first look during pregnancy. There are methods to see in early pregnancy if you have that the fetus has a CMT mutation. Next. And what some parents consider even is to do an artificial fertilization, in vitro fertilization, and use something called pre-implantation genetic diagnostics to exclude that the sort of artificially fertilized embryos that they have CMT. So that's quite costly, and it's definitely not easy for the mothers. But again, it is out there, and some parents go along. They take this on them to avoid their children having CMT. So, of course, a very classic option is also that you use donations of sperm or eggs, anonymous donations, to avoid children having CMT. And a very classic approach also is to an adoption. So things that some families consider, and I just want to list it here so that you see the entire spectrum of what families consider. Next. And to give you also some more detail here, prenatal diagnosis these days uses different approaches, as you know, ultrasound, but also taking small samples from blood or from the placenta to determine if this fetus has CMT mutations. Next. And the pre-implantation diagnostic, the way it works is you have to be willing to do sort of outside of your body an in vitro or the artificial fertilization. And when you do that, then doctors have the option to look at these cells when they still have only 8 or 16 cells. So they're not even fetuses yet. They can look at these cells and study them genetically and determine which one of those to be embryos would actually have a CMT gene and which one doesn't. Next. So also another aspect of all genetic testing is always the question, when I do genetic testing, will this hurt me later in other ways, or my children, my family? And clearly genetic testing is not like taking a blood test. It is something that the genome in your body doesn't change. It never quite changes really. So once you test it once, you have this information for your lifetime. And as I explained in the beginning, you give 50% of your genes, you give down to your children. So actually that information that you get for yourself may very well have an impact on your children. And so these are things to consider, and there are genetic counselors or specialized doctors who can explain all the details for you. It's very good to know that in the United States we have a law called GINA, which protects specifically against misuse of genetic information. So health insurers are prevented to use this information against you. So they cannot refuse coverage, for instance, based on genetic information. Also, employers cannot discriminate against this. You will go one back, please. But there are limits to GINA. The way Congress has written it, it does not regulate how life insurers or other insurers may look at genetic data. Okay, so far, to my knowledge, these insurers don't use genetic information, but that may change in the future. Next. And of course, there's other protection like the HIPAA Act, and you all sign a HIPAA form when you see a doctor. And HIPAA really is to regulate how your personal and private health information is being shared with others. So it's the so-called PHI, the personal health related information that Congress has given very special protection. Okay, next. Next. So, and maybe my last slide here, I believe, is second to last, is to point out that a lot of these studies to advance understanding of CMT and CMT genetics, but also hopefully soon CMT therapies, are possible because we have formed this neuropathy consortium, which includes many, many doctors and genetic counselors and researchers in the United States and also in Europe to work on this together. And next. And that's my final slide showing our group in Miami and acknowledging some of the places that give us support. And I'm here for questions if needed. Thank you.
Thank you very much, Dr. Zuchner. I appreciate you going over all of that. That's a lot of detailed information. We do have a couple questions. This person's asking, and I think you did touch on this, but I'm going to ask. I had exome sequencing completed about 5 years ago, still not received a CMT type other than CMTB. Is this process extremely long to know what type I or my family are? Is this, again, the unknown?
Right.
Okay.
It could be all of this, for sure.
Okay.
The process itself is not necessarily that long, but if you enroll in a research study, research studies don't work on a timetable necessarily. So a clinical test will certainly be done within weeks, but in a research study, it sometimes can take many months to come to a result. Mm-hmm. And also sometimes research studies do not report back to their participants. So it may be worthwhile reaching out, if it's a research study, reaching out to the researcher and asking if there are any news about the test.
Should a person with CMT avoid food and drinks with sorbitol?
That's a good question. We actually don't know the answer yet.
Okay. That's fair.
Yeah, it's a good question. I think that it's probably, I don't really want to give a recommendation here. We really don't know. I tend to think that it probably does not hurt you to have sorbitol sweetener, use it, because it is likely that the sorbitol inside the cells is what is so bad. If it's outside the cell, it may be fine.
Okay. Next, I have CMT type 2 with an unknown genetic factor. Am I correct in assuming that they don't know what's causing my CMT?
Pretty much.
Okay. Okay. And are blood and DNA tests accurate in showing which type of CMT one has?
Yes, they're very accurate. In fact, we think a genetic test that is positive is basically, it defines with great certainty what CMT type you have.
Okay.
And again, I cannot overestimate the importance of this. First, it puts sort of a clear label to the type of CMT you have. Mm-hmm. You can then compare yourself to other CMT patients who have the same label, because the different genes have typically different outcomes. And most importantly, we hope with these new therapies, like gene therapies, you have to have a gene test that is positive. If you don't have this, if you're unknown, unfortunately, very likely you will not be able to participate in these new opportunities.
Okay. Is a DST gene mutation a form of CMT?
Mm-hmm. Yes.
Do you know what type?
It's a type 2, what we call type 2.
Okay.
And it's one of those types, I mean, like many types, that's more rare and there still needs a lot of research to fully understand.
Okay. And I might pronounce this wrong, so I apologize. Does the Cepa 1L2 modifying gene affect other types of CMT?
Yeah, so we have studied this in CMT1A as a modifier. Okay. There we know, I mean, our results, I shall say, support that this is a modifier. Mm-hmm. We have not tested it in other types of CMT, and one reason often is that we don't have enough. So CMT1A is the most common form of CMT. Okay. That we often don't have enough patients for our research studies for the other types. You need a certain number of patients to have sort of statistical power, right, to understand it. So participating in research studies for all CMT patients is going to be very helpful in the long run to improve the situation. Okay.
This person says they had a deep skin tissue biopsy last year. How would this help to identify what family type we have? There's apparently 3 generations of males only.
Yes. So skin biopsy can be very helpful for research. I showed you some research example, mm-hmm. But it sometimes is helpful for diagnostic also, because in the skin, there are also nerve fibers in the skin that there are specialized tests that cannot identify the gene, but it can further narrow what type 1 has.
Okay. Can CMT be attributed to gene breakage in older adults?
Can you repeat this, please?
Can CMT be attributed to gene breakage in older patients?
Well, not sure gene breakage, what
I'm
you
not
mean.
either. Okay, let me see.
No, but I would typically, probably the way I see it, gene breakage, mm-hmm. Probably not.
Okay. Okay. This person said, 10 years ago, my son was told he had type 1A. Mm-hmm. It was then determined that I also have 1A, but to the best of our knowledge, there's no history of CMT on my husband's side of the family, and neither of my parents had it. Yes. Based on what you said about being a sporadic patient, should we seek out additional genetic testing? Are we most likely not type 1A at all?
Well, that's a good example, right? So mm-hmm. It's a mother, it sounds like, mm-hmm. and a son. And CMT1A is a dominant CMT, so that makes sense. Mm-hmm. You would expect that a parent and a child have CMT. Also interesting in this brief description how apparently the son got it much earlier than the mother. Mm-hmm. And that's what I mean, that even though they share the same mutation, the way the disease develops can be quite different. That's why the modifier genes come in. It sounds like this is a pretty solid diagnosis. Uh-huh. There are two gene tests being done on in this family, and they came back positive. So that probably is CMT1A.
Okay. This person says, if I'm reading this correctly, diagnosis of CMT2 without genetic testing. Should I be genetic tested to determine the level of CMT?
Sorry, can you repeat this, Nicole?
This person says they have a diagnosis of CMT2 without genetic testing. Should I be genetic tested to determine the level?
Yeah, I would recommend this very much. CMT2 is a pretty broad category. And as I explained before, knowing your gene is going to be important.
Okay. And I don't know if you would actually know the answer to this question, but I'm going to ask it. How can we find other patients with CMT4B3, especially children?
Yeah, good question. Which touches on a bigger challenge we always have. For these rare forms of CMT, especially, we only often know like a handful of patients.
Wow.
And I think what the person suggests is sort of, how can I find more families around me who also have that?
Mm-hmm.
Right? And so we can sort of join forces and then support research projects or convince pharma companies to help us.
Mm-hmm.
And so the best way probably is to go to research, to patient foundations, like the CMTA, the Hereditary Neuropathy Foundation, the HNF in New York, or the MDA. Of course, here we're talking about MDA.
Yeah, exactly.
The MDA has supported CMT and also my own research for many years. And these associations typically have newsletters, they have registries where patients can connect. And I think it's very valuable to establish networks for your own types of CMT to make progress.
Okay. And I know we're getting to the top of the hour. I just had one more question. This person is 70. They were diagnosed at 40 and beyond. How often should I be reassessed? She sees her neurologist every 6 months, and the last major assessment was done 5 years ago. Without genetic tests, she feels my type may be, is it axonal CMT? AXONAL CMT? So I guess she's asking, how often should she be reassessed?
I think it's something you should discuss with your doctor. There's so many variables from how severe it is and so on. It is certainly a good idea to reassess it from time to time, every 6 months, every year, every 2 years. It's certainly good to follow it. I recommend a genetic test. We find more and more that also individuals with a later onset of CMT have genetic forms of CMT.
Okay.
So yeah.
All right, and then lastly, do you know of any information on the INF2 gene mutation and the progression of CMT?
So INF2, is it?
INF2, correct.
Yeah. So INF is a sort of special type of CMT that seems to very often go along with damages of your kidneys.
Oh, okay.
So there's this connection there, and it's also one of the more rare forms. But what I can say is what you probably already know is to have a close eye on your kidney function.
Okay. All right. Well, that looks like all of our questions after your segment. So thank you very much for coming on again this afternoon.
Absolutely.
And I'm sorry about the technical difficulties earlier, but we're glad we were able to catch up. So thank you very much, Dr. Zuchner.
Thanks, Nicole. Thanks, everybody, for listening in.
And thank you very much. This concludes our CMT Symposium for today. I do want to thank all of our presenters and putting all their presentations together. And thank you for taking time today to join us. Those watching and on-demand recording of this webinar will be available in a couple weeks. And I do hope you found it informative. If you do have questions after the symposium about what you heard or about any other Engage programs, please feel free to visit mda.org/engage. And finally, I want to remind everyone that I will be emailing a brief survey. It's an overview of what you heard and learned about today's symposium. And so we really greatly appreciate your feedback. It might take 5-10 minutes of your time. So if you don't mind filling that out and sending it back, I would appreciate it. So thank you all again for joining, and we look forward to having you join us at the other Engage events we're going to have this year. Have a good rest of your day. Bye-bye.